Hello student, I Dr. R.D. Patil, Head Department of Zoology, Vasantranik Arts, Science and Commerce College, Shada. Dear friends, I already discussed SYBSc Zoology paper first Zoo-401 401 genetics. In this course, we already in our manually at this time the college is start for the early late lockdown after to 15 February as to started at this time in manually the class we discuss the syllabus. विद्यार्थी मित्रांनो तुम्हाला माहितीच आहे ऑलरेडी बऱ्याच 10 ते 11 महिन्याच्या लॉकडाऊन नंतर आपली कॉलेज सुरू झाली त्यावेळेस सेकंड टर्मला वेगवेगळे विषय कुठलेत आपले SYBSc झुलॉजी या कोर्स मध्ये जू 401 याच्यामध्ये सेमिस्टर सेकंड जेनेटिक्स and Zoo 402 semester second zoology paper second that is evolution biology. त्यापैकी जेनेटिक्स या कोर्सच्या संदर्भात आपण सिलेबस रायटिंग करून घेतलं होतं. या कोर्स मध्ये एकत्रित 5 युनिट्स आहेत. तर 5 युनिटचा सिलेबस देखील तुम्हाला मॅन्युअली द क्लास विल बी स्टार्ट ॲट दिस टाइम वी राईट कंप्लीट फॉर द एव्हरी स्टुडंट. प्रत्येक विद्यार्थ्याला आपण लिहून घेतलेलं आहे. त्याच्यामध्ये युनिट पहिलं जे फर्स्ट युनिट इज द इंट्रोडक्शन टू जेनेटिक्स. त्याच्यामध्ये मेंडेल्स वर्क ऑन ट्रान्समिशन ऑफ ट्रेट्स, जेनेटिक व्हेरिएशन, मॉलिक्युलर बेसिस ऑफ जेनेटिक व्हेरिएंट फॉर्मेशन या संदर्भात आपण थोडा ब्रीफली डिस्कस केलं होतं. युनिट नंबर 2 इज द मेंडेलियन जेनेटिक्स अँड इट्स एक्सटेन्शन. मेंडेलियन्स इज द फादर ऑफ जेनेटिक्स. यांनी प्रिन्सिपल ऑफ इनहेरिटन्स, क्रोमोसोम थिअरी ऑफ इनहेरिटन्स, इनकम्प्लीट डॉमिनन्स and complete dominance, polygenic inheritance, multiple alleles, lethal genes, epistasis, pleiotropy, sex-linked inheritance, extra chromosomal inheritance. All kinds of the points are explained briefly in this unit number 2. Unit number 3 is the linkage crossing over and chromosomal mapping. या unit मध्ये आपण linkage and crossing over which type of the particular process are creating in these units. That is recombination frequency as a measure of linkage intensity, 2 factor and 3 factor crosses, definition of gene mapping. हे सर्व आपण याच्यामध्ये डिस्कस करणार आहेत. युनिट नंबर 4 याच्यामध्ये म्युटेशन, व्हॉट इज मीन म्युटेशन and most important roles in the genetics point of view in the mutation. That is different types of mutation means that is chromosomal mutation. In this chromosomal mutation, that is deletion, duplication, inversion, translocation, aneuploidy and polyploidy. Gene mutation, induced version, spontaneous mutation, all kinds of the points are explained in this unit number 4. And last is the unit number 5 is the sex determination. In this course, we discuss the sub point, that is the chromosomal mechanism and methods which are takes place in the particular units are also explained. Dear friends, today we start the unit number first, that is introduction to genetics. विद्यार्थी मित्रांनो, या युनिटच्या संदर्भात ऑलरेडी सिलेबस डिस्कस करताना किंवा Zoom मीटिंग मध्ये today we discuss the particularly briefly idea of this introduction of to genetics in your Zoom meeting. Zoom मीटिंगच्या माध्यमातून या युनिटवरती आपले लेक्चर झालेले आहे. तरी देखील काही मुलांना Zoom मीटिंगच्या माध्यमातून किंवा Zoom ॲपच्या माध्यमातून आपले बरेच मुलं ट्रायबल एरियातले किंवा बऱ्याच एरियांमध्ये नेटवर्कचा प्रॉब्लेम असल्यामुळे पुन्हा मी तेच लेक्चर तुम्हाला युट्यूबच्या माध्यमातून तुमच्या व्हॉट्सअप ग्रुपवर पाठवणार आहे. तरी प्रत्येक विद्यार्थीने हे जे आपलं लेक्चर आहे हे सर्वांनी बघायचं आहे. वैयक्तिक माझे एक्सेल झुलॉजी हे चॅनल्स आहे, या चॅनल्स वरती आपण युट्यूबला जाऊन त्या ठिकाणी सबस्क्राईब किंवा त्याच्यावरती आपण अपलोड करून त्या चॅनलला ऑन करून आपण बघू शकता. असू द्या विद्यार्थी मित्रांनो, द फर्स्ट इन Zoom meetings lectures already discuss about the what is mean genetics. Genetics may be defined as the study of heredity and variation. Genetics is the branch of biology are concerned with the study of genes, genetic variation, and heredity in organisms. Dear friends, genetics is the study of how living things receive common traits from the previous generation. These traits are described by the genetic information carried by a molecule called DNA. म्हणजेच जेनेटिक्स ही दुसरी ब्रांच नसून की ज्याच्यामध्ये अशा प्रकारचे एक सायन्स आहे ऑफ एक्झिस्टन्स फॉर दी स्टडी ऑफ फिजियोलॉजी ऑफ हेरिडिटी अँड व्हेरिएशन, की ज्या ब्रांच मध्ये फिजियोलॉजी ऑफ हेरिडिटी अँड व्हेरिएशन. हेरिडिटी is a particular characters which are transmitted from the one generation to the next generation, and variation to different kind of traits which are also changing the one form to the another form, and get the varied forms are also evolved in particular group. So this is called variation. अशा पद्धतीने हे दोन मेन पॉइंट आपण याच्यामध्ये बघणार आहेत. कारण the science of heredity or genetics is the study of two contradictory aspects of nature, that is heredity and variation. कारण hereditary मध्ये heredity and variation is the two important points are included. The process of transmission of characters from one generation to the next, either by sexual or asexual reproduction. Each and every generation, the characters will be transmitted by the particular process is a reproduction, that is a sexually as well as asexually. In sexual reproduction, really two male and female gametes comes in contact to each other. In asexual, any kinds of the real sex without the sex organs or sexual organs are not participate, but any parts of the body are to form by the next same traits of this organism in the asexual reproduction. That is, heredity is the cause of similarities between individual. Also, as a heredity is the cause of similarities between the individual, the similarity of the offsprings to parents. This is the reason that brothers and sisters with some parents resemble each other and with their parents. Variation is the cause of difference between individuals, the difference between the parents and offsprings, and between the offspring themselves. This is the reason that brother and sister who do resemble each other to still unique individual. Thus, we have to trouble in recognizing the difference between sisters. Example, and even identical twins are recognized as distinctive individuals by their parents and close friends. So this is a particular events to get by the heredity and variation. In genetics, the genetic information is carried by DNA. A gene is a hereditary unit consisting of DNA that occupying a spot on a chromosome and determines a characteristics in an organism. Gene is the constituents part of the DNA. Dear friends, we know about what it means genetics. Genetics is the field of science that examines how traits are passed from one generation to the next. And organisms, genes, snipes of DNA that are the fundamental units of heredity, control how it looks, behaves, and reproduces. We already discussed the genetics to genes. We also in your Zoom's lectures. The term genetics was coined by capital W Bateson 1906 and was derived from Greek word gene, means grow into or to become. That is particular meanings of this gene or genetics. The term genetics was coined by W. Bateson 1906. Genetics may be defined as the science of existence for the study of physiology of heredity and variation. We already discussed the point in the first lectures. That is, first, what is the genetics? The branch of biology. Genetics is the branch of biology that deals with the heredity, especially the mechanism of hereditary transmission and the variation of inherited characteristics among similar or related organisms. Means that is the particular to that transmit by the identical characters, or also uh, uh, to process of the transmission by the character from the one generation to the next generation. In a division point of view or a classification point of view, the genetics are divided by the four main branch, that is the classical genetics, molecular genetics, population genetics, and quantitative genetics. All kinds of the branches of the genetics in your study point of view. Next, we already discussed in the gene, a small molecule of DNA, biological unit of heredity. Gene is nothing but the biological unit of the heredity. Gene hold the information to build and maintain their cells and pass genetic traits to offsprings. It is in cells, gene is a protein of DNA. In simple way, it is called as a protein of the DNA. सो इन जेनेटिक्स पॉइंट ऑफ व्ह्यू डिस्कस. विद्यार्थी मित्रांनो, आपलं जेव्हा मॅन्युअली वर्ग स्टार्ट झाले त्या टायमाला मी 2 ते 3 लेक्चर मध्ये जेनेटिक्स या सब्जेक्ट्स मध्ये येणाऱ्या वेगवेगळ्या ज्या टर्मिनॉलॉजिस्ट आहेत त्या सर्व तुम्हाला मी रायटिंग करून दिलेल्या आहेत. त्यापैकी ब्रीफली ऑलरेडी अगेन टू एक्सप्लेन यू, त्याच पुन्हा मी तुम्हाला शॉर्ट मध्ये अजून एक्सप्लेन करून देतो. टर्मिनॉलॉजीज in genetics. The term genetics was coined by William Bateson in 1905. First is the gene. The term gene was coined by Johnson in 1909. Gene is the hereditary determining factors. Gene consists of continuous segment of DNA. In eukaryote, the gene occupies in specific position on the chromosome called locus or loci. So this is a William Bateson's and also as a Johnson to found by the genes. This is a chromosome, this is a DNA and genes also in this diagram. So in a genetic point of view, second term is the allele, also called as the allomorphous. Alleles are the alternative forms of gene which occupy identical loci on homologous chromosome. Allele control the contrasting characters of the same trait. Usually alleles exist two different forms, that is dominant allele and recessive allele. We also indicating in the chromosome pair to that is a allele as a specific form. In third, dominant and recessive, we mainly the most important these two words or two meanings are used by the, that is the dominant characters or recessive characters. The dear friends, we first known about the what is meant dominant. Dominant alleles will be always expressed. Recessive alleles will express only in the absence of dominant allele. दोघांमधला हा फरक आहे डॉमिनंट रेसिसिव्ह मध्ये. डॉमिनंट allele mask or suppress the expression of recessive allele. Dominant alleles are classically symbolized with capital letters. Capital letters दाखवले जातात, इंटिमेशन केले जातात. Recessive alleles are symbolized with small letters. For example, in these traits, that is the height, these characters also indicating by the tall and dwarf. Tall means त्याला उंच म्हणतो आपण, डॉर्फ मीन्स हाईट कमी. हाईटच्या बाबतीत उंच असला, हाईट जास्त असला, हाईट कमी असला, त्याच्यामध्ये टॉल, डॉमिनंट टॉलचं आपण इंटिमेशन करण्याकरता कॅपिटल टी, रेसेसिव्ह डॉर्फचं स्मॉल टी आपण त्या ठिकाणी ते वर्ड्स युटिलाइज करतो मेकॅनिझम करण्याकरता. So this is the next term is a genotype and phenotype. This type of the meanings also already explained in many more time in the mechanism of the genetic reactions as well as the genetic terms to indicating by the different kinds of the law. At this time we include the two terms, that is genotype and phenotype. It is most important term. So each and every student we known about what is mean genotype, the genetic makeup constitution of an organism. Genotype is nothing but the genetic makeup we also in the internal to animal organism, and phenotype is nothing but the physical features or appearance of an organism. We also including to morphological behavior. म्हणजे एकाच वेळेस the genetic genotype and phenotype. Phenotype is nothing but a morphological attitude, physical attitudes to get by the phenotype. Genotype also has a internal constituents or genetic makeup in the of an organism. Phenotype is the expression of genotype. Phenotype is the produced not only by the genotype but also by the interaction between genotypes and environmental factors. Trait, that is height. Height चे character जर आपण घेतलं त्याच्यामध्ये phenotype tall and dwarf. Genotype मध्ये capital TT or capital T, small t and small tt. So this is an intimation by the, in phenotypic point of view, capital TT or small tt. But in genotypic point of view, capital TT, capital T, small t and small tt. This is a pure dominant, pure recessive. It is a heterozygous dominant. So these all kinds of traits are also intimating in these P plants. That is, this also discuss about the phenotype and genotype. Different alleles may be produced differences in the observable features or phenotypic of an organism. The set of the alleles for a given trait carried by an organism is called a genotype. This is a observable feature is a phenotype, and this is a carried by an organism is called as a genotype. Knowing gene control the phenotype and genotype. What is the chemical natures of gene? By the 1920s, scientists were aware that proteins and DNA were the major chemical components of the chromosomes. That is the DNA were the major chemical components of the chromosome. That is DNA and proteins. Next, that is the genotype and phenotypic. We already discussed to genotypic is genetic makeup of an individual. It is determined by the alleles present for each trait. Phenotype is a physical appearance. This is genetic makeup. This is a physical appearance of the traits. It is the expression of the genotypes. So this is a characteristic features which is seen by the diagram by the genotype and phenotypic. बऱ्याच टायमाला आपण म्हणतो की in your upstream जेव्हा आपण आपल्या घरामध्ये फॅमिली आपण आई वडिलांचे 50% कॅरेक्टर हाफ कॅरेक्टर्स फ्रॉम युअर मदर हाफ कॅरेक्टर्स फ्रॉम युअर फादर अँड गेट बाय द ट्रेड्स दोघांचे 50% कॅरेक्टर जरी आलेले असले तरी बऱ्याच वेळेस आपण आपल्या घरामधले मेंबर बहीण किंवा भाऊ हे आपण बऱ्याचदा म्हणतो की बहीण ही आईसारखी किंवा बहीण ही वडिलांसारखी मी आईसारखा आहे असं आपण एकमेकांना आपल्या घरात आपण ट्रेड्स बघतो. ऍक्च्युअली जो जर एखाद्या वेळेस बहीण वडिलांसारखी दिसत असेल तर त्या ठिकाणी वडिलांचे कॅरेक्टर डॉमिनंट्स आहेत आणि आईचे कॅरेक्टर रिसेसिव्ह आहेत. आणि मुलगा जर आईसारखा दिसत असेल तर वडिलांचे कॅरेक्टर त्याच्यामध्ये रिसेसिव्ह आहेत, आईचे कॅरेक्टर डॉमिनंट्स आहेत. सो दिस इज अ पर्टिक्युलर traits to get by the genotypic and phenotypic point of view. That is in the homozygous condition in which next term is homozygous, a condition in which both the members of allelic pair in the homologous chromosome are identical. That is a tall. We also traits of capital TT, dwarf, small tt. So this is a capital TT is a homozygous, small tt is a homozygous. But to get by the mixing capital T and small t, it is called heterozygous. In next term, condition in which the members of an allele pair in the homologous chromosome are not identical. One dominant and one recessive allele are, that is the example tall. One is the capital T is the dominant, small t is the recessive. This trait is the heterozygous. Next is the hemizygous. In a condition when genes are present only in one copy. Gene X chromosome is the male, have a X and Y chromosome. Gene on Y chromosome only one Y chromosome in males. So this is a hemizygous. Dominance, the ability of an allele to express itself phenotypically both in homozygous capital TT and in heterozygous capital T small t condition. दोघांमध्ये त्या ठिकाणी डॉमिनंट कॅरेक्टर आपल्याला दिसत असतात. ॲबिलिटी रेसिसिव्हनेस मध्ये जर बघितलं, द इनएबिलिटी ऑफ एन allele to manifest its phenotype in heterozygous capital T small t condition. त्या ठिकाणी त्याच्या प्रकारची ऍक्टिव्हिटी आपल्याला रेसिसिव्ह दिसते. नेक्स्ट दिस इज अ hybridization and hybrid. Hybridization, the process of crossing two genetically different individual, that is homozygous tall to indicating by capital TT, homozygous dwarf to indicating the small tt. By the capital TT homozygous to get the T is the one gamete in homozygous dwarf, small t is a gamete to mixing at the fertilization to call as a capital T and small t. It is called a heterozygous tall. Hybrid, the progeny of hybridization is called a hybrid. जी प्रोजेनी आलेली त्याला आपण हायब्रिड प्रोजेनी आपण म्हणतो. वन capital T is homozygous tall, small t is a homozygous dwarf and combined to form as a heterozygous tail. So this is a hybridization or hybrid. In genetics point of view, the two important terms, that is a what do you mean monohybrid ratio and dihybrid ratio. आपण already discussed in Zoom meeting की मोनोहायब्रिड रेशो म्हणजे काय, डायहायब्रिड रेशो म्हणजे काय. फर्स्ट टू मोनोहायब्रिड, an organism which is heterozygous which repeat to only one pair of allele at a locus under study. Only one character that can आपल्याला दिसतो, that is tall and dwarf, one character only to transmit. In dihybrid, an organism which is heterozygous with respect to two pairs of alleles at two loci, with one pair of allele at a locus, only one locus under study. या ठिकाणी two pairs of alleles at two loci under study. That is, yellow is a color, round, yellow round seed. Yellow is the color, round seed. Round is the shape, means color and shape. Green is the color, winkle is the shape. This is the two characters are transmitted to this trait, two pairs of alleles at two loci under study. So this type of the condition is called a dihybrid. Dear friends, विद्यार्थी मित्रांनो, we also मोनोहायब्रिड क्रॉस, डायहायब्रिड क्रॉस. जसं आपण मोनोहायब्रिड डायहायब्रिड म्हणजे काय ते बघितलं, क्रॉस करताना a cross between two individual organisms which differ from each other with respect to one pair of allele under study. That is capital TT tall, small dwarf, small tt. In dihybrid, that is a cross between two individual organisms which differ from each other with respect to two pairs of allele under study. That is yellow round seed, green wrinkle seed. That is a yellow capital YY, round shape capital RR. Green small YY, wrinkled shape small RR. So this is a two characters also crossing two pairs of the alleles under the study of the dihybrid cross. That is F1 and F2 generation. That is F is a filial meaning son. F2 is the first generation progeny of hybridization. F2 is the progeny of hybrid F2. F2 is the progeny of hybrid F1 when it is hybridized with any of its parents. Means that is a capital TT, small tt to get by your gametes, capital T and small t to intimates as a specifically in point of view this tt small also to got by the specific process in this way. That is a F1 generation is a capital T small t cross to recessive parents small tt. This gamete is the capital T small t, is the only one gamete, that is small t. F2 generation is the capital T small t and small tt. So this is a this is F1 generation. In F2 generation is the capital T small t. This is a heterozygous dominant. This is a homozygous recessive. So this is the particular points are also indicating. In next, that is a reciprocal cross. That is a to receive reverse crosses in which the sexes of the parents are interchanged. That is a tall is a male, dwarf is a female. This is a dwarf is a male, tall is a female. Alternative cross is a reciprocal. If the traits are autosomal, the reciprocal cross always yield same results. In the traits or on sex chromosome, or reciprocal cross gives different results. So this is specifically as a cross alternately to you by the in reciprocal cross. That is the back cross. In back cross, the cross of F1 offsprings with one of its parent. If F1 is crossed with dominant parents, all progenies F2 will be dominant. If F1 is crossed with recessive parents individual, with both phenotype will be appear on equal proportion. The ratio of progenies produced during back cross is called a back cross ratio. So this is a particular events. Next is the test cross. In test cross is a type of back cross in which the F1 progeny is crossed with its double recessive parent. Test cross is used to determine whether the individual exhibiting dominant characters are homozygous or heterozygous to detect the genotype of F1. To, for example, point of view, capital TT cross to small tt to gametes in capital TT from capital T in small tt small t. F1 generation is a capital T small t to cross by also recessive small tt, and in a capital T small t is two gametes capital T and small t. in a small, both small tt only as one gamete, small t in F1 generation to percolate by the specific processes takes place. Next is the in point of view, that is the Mendelian laws. In the specific to discuss about the in Mendelian laws, first we know about the Mendelian law, that is monohybrid dihybrid, but we mostly the John Mendel 1822 to 1884 in Austrian monk was the founder of the modern science of heredity. He is therefore often referred to as the father of modern genetics. Mendel evolved an entirely new approach to concept of the transmission of characters. The contribution of Mendel's work to genetics is called Mendelism. मेंडेल सायंटिस्ट त्या ठिकाणी जेनेटिक्स मध्ये काम केलं त्याला मेंडेलिझम म्हणतात. ही परफॉर्म्ड अ सिरीज ऑफ एक्सपेरिमेंट्स विथ पी प्लांट्स इन द गार्डन. हिज वर्क कंटेन्स इनहेरिटन्स ऑफ कॅरेक्टर्स इन 22 व्हरायटीज ऑफ गार्डन पीज. मी सकाळी अर्ली झूम मीटिंगचा लेक्चर मध्ये विद्यार्थी मित्रांनो तुम्हाला एक्सप्लेन केलं की जेनेटिक्स किंवा मेंडेलने जेनेटिक रेशोचा अभ्यास करण्याकरता किंवा जेनेटिक इनहेरिटंट कॅरेक्टरचा अभ्यास करण्याकरता प्लांट्स या ग्रुपमध्ये पी प्लांट घेतलेला आहे आणि ॲनिमल ग्रुपमध्ये ड्रोसोफिला मेलॅनोग्लास्टर मीन्स फ्रूट फ्लाय त्या ठिकाणी घेतलेली. याचं कारण मी सकाळी बोललोच तुम्हाला की पी प्लांटच्या जवळजवळ 22 व्हरायटी आहेत आणि पी प्लांट मध्ये क्रोमोझोम नंबरिंग इज 11, 11 क्रोमोझोम त्या ठिकाणी असल्यामुळे त्यांचे ट्रेट जे आहेत इझिली फाइंड आउट करता येतं. त्याच पद्धतीने ड्रोसोफिला मध्ये देखील कमी पेअर्स ऑफ क्रोमोझोम असल्यामुळे त्यांच्यामध्ये विच क्रोमोझोम्स आर चेंजिंग as a particular variation, hereditary mutation to get by the counting easily to find out. त्या ठिकाणी बघण्याकरता त्यांनी घेतलेलं आहे. जॉर्ज मेंडल though his work on pea plants discovered the fundamental laws of inheritance. मेंडेलवरती त्यांनी जे एक्सपेरिमेंट केले त्यांना लॉज ऑफ इनहेरिटन्स म्हटले गेले. ही deduced that genes come in pairs and inherited as distinct units, one from each parents. Mendel tracked the segregation of parental genes and their appearance in the offsprings as dominant or recessive traits. He recognized the mathematical patterns of the inheritance from one generation to the next generation. अशा पद्धतीने त्या ठिकाणी त्या मोड्सला ते मेंडेलने ते रेशो मांडलेले आहेत. मग मेंडेल वाय मेंडेल्स टू सिलेक्शन ऑफ एक्सपेरिमेंटल प्लांट्स. मेंडेलने सिलेक्शन ऑफ एक्सपेरिमेंटल प्लांट्स साठी पी प्लांट्स का घेतलं बघ त्याच्यामुळे म्हटलेलं त्यांनी वेन मेंडेल स्टार्टेड हिज हायब्रिडायझेशन एक्सपेरिमेंट्स ऑन द इनहेरिटन्स ऑफ कॅरेक्टर्स इन 1857. 1857 मध्ये त्यांनी पहिल्यांदा सिलेक्शन एक्सपेरिमेंटल प्लांट्स वरती सुरुवात केली मेंडेलने. ही हॅड अ क्लिअर कन्सेप्ट अबाउट हिज टास्क ऑफ फाइंडिंग आउट हाऊ डेफिनाइट अँड फिक्स्ड व्हरायटीज ऑकरिंग विदिन अ पेसिस ऑफ रिलेटिव्ह टू वन अनदर. त्या ठिकाणी त्यांनी ट्रेट शोधून काढल्यानंतर त्याच्यामध्ये कॅरेक्टरिस्टिक्स ऑफ व्हेरिएशन. It means experimental plants must be necessarily processes constant differentiating characters variation, and the hybrids of such a plant must during the flowering period be protected from the influence of all foreign pollen grains or to easily capable of such protection. Moreover, he suggested that the hybrid of experimental plant and their offsprings should suffer to mark the distance in their fertility. अशा पद्धतीने त्याच्यामध्ये सक्सेसिव्ह जनरेशन कसे होतात ते केले गेलेले. ही फाउंड द प्लांट ऑफ फॅमिली ऑफ लेग्युमिनेसी. ही प्लांट स्टडीला घेतल्यामुळे पी प्लांटची बॉटनिकल फॅमिली लेग्युमिनेसी आहे. सच ॲज पीज अँड बीन्स मोस्ट सुटेबल मटेरियल फॉर द हायब्रिडायझेशन एक्सपेरिमेंट्स बिकॉज दीज प्लांट्स. त्या प्लांटमध्ये असे काय कॅरेक्टरिस्टिक फीचर्स किंवा गुणधर्म आहेत की ज्याच्यामुळे मेंडेलला एक्सपेरिमेंटल लॉ ऑब्झर्वेशन करण्याकरता त्याची गरज पडली का घेतला असेल. त्याचे काही महत्वाच्या गुणधर्मांच्या आधारावर त्याच्यातलं पहिलं आहे व्हेरी इझी टू कल्चर टू ओपन ग्राउंड ऑन द पॉट्स. वाटाणा प्लांटला आपल्याला सहज ओपन ग्राउंडला कल्चर करता येतं. सेकंड आहे हॅड शॉर्ट ग्रो पिरियड अँड लाईफ सायकल. त्याचं लाईफ सायकल लवकर ग्रो होऊन लवकर त्याचं सर्व ऑब्झर्वेशन अभ्यास करून त्याचं लाईफ सायकल पूर्ण करता येतं बघता येतं. हॅड सेल्फ पॉलिनेटिंग फ्लोअर्स ऑफ पिक्युलर स्ट्रक्चर. सो जवळजवळ स्वतः सेल्फ पॉल्युशन करून घेतात त्यामुळे त्यांच्यामध्ये फर्टिलायझेशन लवकर होऊन फ्लोरिंग वगैरे काय त्याचा अभ्यास आपल्याला लवकर करता येतो. हॅड कॉन्ट्रास्टिंग हेरिडिबिलिटी कॅरेक्टर्स. त्याच्यामध्ये विशिष्ट प्रकारचे हेरिडिटी कॅरेक्टर लवकर आपल्याला बघता येतं. आणि पाचवं आहे माइट प्रोड्यूस फर्टाईल हायब्रिड्स ऑन आर्टिफिशियल क्रॉस पॉलिनेशन. त्याच्यामध्ये फर्टाईल हायब्रिड जी कॅरेक्टरिस्टिक फीचर आहे ते आर्टिफिशियल क्रॉस पॉलिनेशन करून आपल्याला बघता येते. मेंडेल फाउंड edible pea Pisum sativum plant is best material for his hybridization experiment because its various availability 34 species 34 species त्यांनी त्या ठिकाणी घेतल्या. He shows that many differentiating characteristics are present in the plant such as follow. त्याच्यामध्ये कुठले कुठले पॉइंट क्रॉसिंग करण्याकरता आपल्याला बघावं लागला. हाईट ऑफ द प्लांट दॅट इज द टॉल अँड डॉर्क, द कलर ऑफ फ्लोर रेड अँड व्हाईट, द शेप ऑफ द सीड राऊंड अँड विंकल, द कलर ऑफ कॉटिलिडॉन फुलाचा मागचा जो कॅलिक्स करोला त्याचा कॉटिलिडॉनचा कलर कसा असतो येलो और ग्रीन, द पोझिशन ऑफ फ्लॉवर ऍक्झिलरी और टर्मिनल कुठे लागलेला आहे बुवा, द कलर ऑफ अनराईप पॉड पिकलेला त्याला आपण पिकलेला फळ किंवा अनराईप ना पिकलेला फळ लाईट टू डार्क ग्रीन और येलो ते केलं जातं. आणि सातवा कॅरेक्टर आहे शेप ऑफ पॉड इन फ्लॅटेड और कॉन्ट्रॅक्टेड. त्याचे कसे असतात हे 7-8 कॅरेक्टर त्याच्यामध्ये बघून पी प्लांट मध्ये व्हेरिएशन मेंडेलस लॉ ऑब्झर्वेशन करण्याकरता अभ्यास करण्याकरता इझिली असतं. ऑलरेडी डिस्कस इन द झूम लेक्चर. झूम लेक्चर मध्ये मी तुम्हाला त्या संदर्भात डिस्कस केलं होतं. पुढचा पॉईंट त्याच्यामध्ये आपण बघू शकतो विद्यार्थी मित्रांनो, मेंडेल्स प्रोसिजर. मेंडेल्सने या ठिकाणी फॉर गेटिंग एक्झॅक्ट रिझल्ट ऑफ ब्रीडिंग ऑफ एक्सपेरिमेंट साठी त्यांनी इट हॅज अ नेसेसरी टू मेंडेल अ प्युअर व्हरायटी ऑफ पी प्लांट्स. त्या ठिकाणी त्यांनी जनरेशन फिनोटाइप पेरेंट पी जिनोटाइप कॅपिटल टी टी स्मॉल टी टी F1 जनरेशन capital T small t, F1 cross to F1 capital T small t and get the 5 traits in the F3 generation. We already discussed in the early tomorrow by in a morning lectures of the Zoom to detail discuss this point in the board. Next, to start the particular ratio, that is a law monohybrid ratio, it is also called as the law of dominance. This is a particular 3 important laws we discuss about in genetics of these units. That is, first is the law of dominance or monohybrid, second is the law of purity or also called the law of segregation, and third is the law of independent assortment. These 3 law is most important for the in this units in study point of view. Dear friends, विद्यार्थी मित्रांनो, त्यापैकी आपण पहिला जो लॉ आहे लॉ ऑफ डॉमिनन्स और मोनोहायब्रिड्स. We know about the first, that is the cross between the organism differing in single pair of contrasting character is known as the monohybrid cross. Mendel made a monohybrid cross between tall and dwarf pea plants. In first F1 generation, only tall pea plants appeared, but when in F2 progeny were allowed to self-fertilized, both tall and dwarf characters appeared in the second F1 generation. This shows that in F1 hybrid, the characters of tallness dominates or conceals the characters of dwarfness, and so the characters of dwarfness could not express itself in the first generation. The characters which expressed in F1 generation are called by Mendel as dominant characters. जे F1 generation मध्ये कॅरेक्टर येतात त्यांना डॉमिनंट कॅरेक्टर्स म्हटले, while the characters which are remained unexpressed called as recessive characters. F1 जनरेशन मध्ये एक्सप्रेस होणारे नंतर दुसरे न एक्सप्रेस होणारे जे एक्सप्रेस होणारे कॅरेक्टर्स आहेत त्यांना डॉमिनंट कॅरेक्टर्स म्हटलेले, आणि दुसरे जे अनएक्सप्रेस कॅरेक्टर्स आहेत त्यांना recessive dominance characters आपण म्हटलेले. Therefore, according to the Mendel, the laws of dominance which state that the crossing between pure म्हणजे homozygous organisms for contrasting characters of the pair, only one characters of the pair is appear in the first generation. That is, parents P homozygous tall crossed by homozygous dwarf. The gametes capital T small t. Also in F1 generation capital T small t, it is called heterozygous tall, and in F1 generation cross to F1 generation, that is the capital T small t cross with the capital T small t, that is in F2 generation capital T capital T small t capital T small t and small t, that is as a 3:1 ratio. That is, capital TT as the homozygous dominant, capital T small t is the heterozygous dominant, capital T small t is the heterozygous dominant, and both are the small tt as a homozygous recessive. So this is a specific to get by the in a law of dominance. We already discussed in the monohybrid cross, a method of determining The inheritance pattern of a trait between two single organisms. A cross between parents who are uh, true breeding for a trait that is both the homozygous or one allele of the gene are example capital A or small a. That is also in the capital TT for tall, small tt for dwarf, as which A is the dominant allele for a trait and small e is the recessive allele for the same trait where we discussed by the capital TT and small tt. That is Mendel's law of segregation. Monohybrid cross means the statement point of view, one pair of contrasting traits are brought together in a cross. We already discussed the uppermost slides. That is the Mendel cross true breeding. Tall TTP plants and also small is breeding. Drop plant TT we also crossing. We already explained by the parents F1 generation, F2 generation. That is the in law of dominance point of view, the trait which has appeared in F1 was called dominant, while the traits which are disappeared was called as a recessive. We already discussed by the later on to description. The F1 plants were self-fertilized and offspring produced were called as F2 generation. Three types of plants were present in F2, that is pure tall, hybrid tall, and dwarf. We already discussed in this schematic representation by the Zoom meeting and the board. Next is that is the graphical representation by already discussed. This is the law. P in parent first, that is tall and dwarf. F1 is all tall. If tall plants are represented by the capital TT and dwarf plants by small tt, then the above cross can be shown as P1, that is capital TT, small tt. These four two gametes, that is two gametes, which crossing in F1 generation is the capital T small t. We already discussed the next slides. That is the in P2, that is a capital T small t, capital T, that is F1. Both F1 cross 2 by these gametes, capital T small t, capital T small t, that is formed by the capital TT, capital T small t, capital T small t, small tt. That is the phenotypic ratio is the tall and dwarf 3:1 ratio. In genotypic, that is a 1:2:1. That is pure tall, also as a hybrids and also the homozygous to get by the conducting you. This is a monohybrid law of dominance. In this point of view also as a law of dominance, the F1 generation capital T small t, this cross two gametes capital T small tt and formed by the capital T small capital T small t capital small t small tt. So this is schematic representation by to get by the 3:1 ratio, that is a 1:2:1 to get peculiar features of the schematic representation by the law of dominance. That is also this 3:1 ratio as a phenotypic ratio. That is genotypic is a 1:2:1, that is a pure tall, hybrid tall, and dwarf. So this is a specific ratio. Dear friends, in this point of view, the law of dominance we also or monohybrids are completely explained in this lecture. Dear friends, in second lectures to explain the law of purity as a law of segregation and law of independent assortment. Dear friends, we already discussed briefly in Zoo lectures on Zoo app to early tomorrow lectures to same lectures also get by the many more students are not attending in the Zoom meeting, so we also explain the YouTube on YouTube channels to get by the manually as well as online to this lecture. Dear friends, all student has a stay home, stay safe, because in your present situation of the COVID-19 is a very severe pandemic. So dear friends, stay home, stay safe. Thank you, thank you, thank you.